Hi, science friends. I hope you all had a nice Thanksgiving and are ready to be back and grind it out for 3 weeks before we get into Christmas break. So what we're going to be doing for the next 3 weeks is wrapping up this unit that we started right before Thanksgiving. So we did enzymes. We're going to finish up enzymes really today, and then we are going to start I guess we'll finish up enzymes tomorrow. You'll have a little lab to do with it with toothpicks. So if you haven't already gone to school and got your materials, make sure to do those to get that. Those were there for you to pick up last week. So make sure to pick up those materials at school. And then we've got cellular respiration and photosynthesis. So that's what we're going to be working on. So let's go ahead and review. Explain the effect that enzymes have on activation energy. So you should say that enzymes lower the activation energy. So remember, activation energy is the energy that's needed to get a reaction started. So in our lesson, I talked about hydrogen peroxide and how that breaks down very slowly over a long time. But if you add a catalyst or an enzyme to that reaction, then it would happen a lot quicker. So enzymes fall in the category of macromolecules as a protein. They are proteins, okay? What is a substrate? A substrate is what an enzyme acts on or what binds to an enzyme to cause a chemical reaction. It's what it works with, okay? And then what is meant by induced fit? So induced fit is how an enzyme and a substrate kind of mold to fit together. They interact once they get together and mold to make an induced fit. So that's what that means. So today we are going to talk about some different ways that enzymes are regulated and also the effect of the environment on enzymes. So enzymes are proteins, and you should remember about proteins that they have these 4 levels of structure: the primary, secondary, tertiary, and quaternary structures. And that if proteins are not in their ideal environment, then they unravel or lose their shape, and that's called denaturing. And so the ideal environment for enzymes and proteins is they have to have a good temperature, an ideal temperature. They have to have an ideal pH. That's going to affect how they function. Also, you may want to add to this list salinity, how much salt is in the solution. That will affect the functioning. Now, not all enzymes and proteins, for that matter too, that aren't enzymes, have the same pH requirements or temperature requirements. Some of them can work in really hot environments, some of them in very cold environments, some of them in very acidic environments, some of them very basic environments. So you shouldn't say that a pH of 7 or a temperature of 98.6 is ideal for all enzymes, because that's not the case. There's lots of different enzymes depending on the organism, depending on where they live in the organism. And so they have an ideal pH and temperature and salinity range that they work best in. Outside of that, they're not going to automatically denature if they get a degree off, but they don't work optimally, okay? There's an optimal or an ideal range. And then if they get really far outside of it, then they can and will start to denature. So enzymes are also affected by the concentration of the enzyme and the concentration of the substrate. And that's what we're going to demonstrate a little bit more with the lab that you're going to do tomorrow with the toothpicks. So let's go ahead and get started. So cofactors. Cofactors are things that work with enzymes. They're non-proteins that help catalyze a chemical reaction. So there's something that work along with enzymes that catalyze a chemical reaction. Sometimes you can also have a coenzyme. So that's something that's biological, that's organic, such as a vitamin. A vitamin can be a coenzyme that it's just a specific type of cofactor. Again, helps with chemical reactions. So that's why vitamins are important, and we take them for our health and the health of our body, because they help our chemical reactions and our metabolic functions to carry out appropriately. Okay, so enzyme action. Enzymes, there are a couple types of ways that enzymes can be kind of turned off and turned on. And so one way is through competitive inhibition. And so we see competitive inhibition right here. So normally, in a normal binding, you have the substrate, and it binds here in the active site of the enzyme, and they fit together nicely. They look like they fit, and then they have that induced fit where it's going to kind of hug around the substrate, the enzyme will. But in competitive inhibition, you have another molecule that blocks the substrate from binding to the enzyme. Okay, it blocks it. It doesn't change the enzyme in any way, but it blocks it, and so it competes and therefore inhibits the binding of the enzyme and the substrate together. So that's competitive inhibition. You have to know these terms, okay? Non-competitive inhibition, on the other hand, is when you have this non-competitive inhibitor molecule down here that binds to the enzyme. So often remember from cellular communication, when you bind, you change a shape, right? So this enzyme binds, and it causes this to change its shape. And so while the substrate can still bind, it perhaps doesn't bind as effectively. And so it makes this reaction less effective, okay? So it can still work, although sometimes it may not, but if it does work, it will make it less efficient, okay? Now, allosteric regulation, so these are all, again, ways that enzymes are regulated. Allosteric regulation, you can have allosteric activation and allosteric inhibition. So in an activator, when an activator binds, it opens all of these active sites up so that they can receive substrates. It stabilizes them in an active form, okay? When you have an inhibitor that binds, it stabilizes them in an inactive form. A substrate can't get in and bind there, and so that inhibits the enzyme and the substrate from working. Okay, so allosteric regulation, both of these are called allosteric regulation. We've got activation and inhibition. When a protein's function at one site is affected by the binding of a regulatory molecule to a separate site. And that's what's happened here. You have the activator or the inhibitor that's bound at a different site that affects the activity of that enzyme or that protein. Okay? Now, cooperativity. Cooperativity sometimes happens when you have enzymes like these, which are also very similar to these, that have multiple subunits. So remember, in our proteins, we have those primary, secondary, tertiary, and quaternary structures, and that quaternary is taking multiple polypeptide subunits. And so that's what these are. This is like 4 different structures. These are 4 different subunits that are all the same, but at different subunits of the enzyme. And so cooperativity, when you have a substrate that binds at just this one site, notice how that shape changed from the inactive form to the active form. It stabilized that active form and makes it more likely that a substrate can bind in the other site. Okay, so substrate binding in one active site triggers a shape change in all of the subunits, increasing the activity of the other active sites. Okay, and that's what cooperativity is. All right. So, feedback inhibition. We talked about feedback inhibition at the very beginning in our very first unit. And so what happens in feedback inhibition? This example I want you to tell me in just a minute. Look at this and tell me, is this negative or positive feedback? We have an end product that binds to an allosteric site and closes the active site up. And so it can't complete the reaction. This is a negative feedback loop. Okay? So what we have here is we have our initial substrate, which is threonine, okay? And it binds to the active site of the enzyme. Ultimately, we are trying to, in this cellular communication pathway, this signal transduction pathway, these are the transduction steps. We are trying to catalyze the production of isoleucine, okay? And so when isoleucine builds up, it actually binds to the allosteric site of the enzyme and it inhibits its own production, okay? It inhibits its own production. So when there gets to be excess isoleucine and it's not needed in the cell as fast as it's being used, it'll bind to the enzymes and cause the production of it to stop, okay? Now, if isoleucine is needed to be used again, then it will be pulled off of here and used, okay? And so thus this shape change will happen and the substrate can bind and will turn this reaction, this pathway back on, okay? And so this is negative feedback. It slows its own synthesis or it increases its own synthesis to make it when it's needed. So what is isoleucine? You may ask. It's an amino acid that's used to make proteins, okay? So that's very important. So again, just revisiting feedback inhibition here. So that's kind of a short lesson. That's all I've got for you today. You have an enzyme worksheet in your binder, so go ahead and complete that and upload it to Google Classroom and let me know if you'll have any questions. You'll have a great day.